recording. Okay, so whenever you want to start, Jack.
Yeah, I'm ready. So let me just share the screen, and let me start by giving you a very quick recap of what has been going on until now. This is the third part of this four-part series of lectures on quantum biology. In the first lecture, I gave you a very broad brush overview of quantum physics from the beginning until roughly today, without too many details, but the general ideas and applications. In the second lecture on Tuesday, I discussed the pros and cons of quantum effects in biology in general, the time scales, the length scales, the energy scales, where things overlap, and also towards the end I started talking about some electromagnetic effects, which are somewhat related. It's not the same, could be classical, but at high enough energies, electromagnetic waves become quantized and above thermal threshold. So these effects become meaningful within the context of biology. And in particular, I ended up with the very attractive theory of long-range coherence in living systems proposed by Herbert Fröhlich in the 70s. And the key aspect of it was that there are potentially collective excitations in living systems, electromagnetic in nature, but quantized, that create a large Bose-Einstein type condensate, which would allow us to understand how synchronization occurs at the cell level and above. And so today I will get deep into the weeds of what quantum biology actually is as we know it today. And then it'll be a bit technical, so I will not expect everybody to understand everything here, because it requires a little bit of quantum physics, but you will get the gist of it. And on Tuesday next week, which will be the last lecture in this series, I would like to invite as many people as possible, because this will be probably the most exciting talk of the series on how quantum effects may explain things happening in our brain. So we get into the interesting effects in neuroscience, and in particular concepts like memory, anesthesia, and if time permits, maybe some aspects of consciousness and even psychiatry. But today it's kind of down to earth. So I will start with putting this in presentation mode. So we'll enter this gently, and this connects to the previous talk about electromagnetic effects in living systems, potentially quantum, but not necessarily. And this cartoon is the origin of this cartoon is from the website that Professor Günther Albrecht Bühler from Northwestern University in Chicago, Evanston, in fact, put together with a very, very intriguing website that I encourage you to visit. I think you can Google it by putting in intelligent cell or cell intelligence, in fact. So he argues that that cells are intelligent. They have some innate intelligence, maybe proto-intelligence, in terms of their behavior, deliberate actions, seeking food and favorable living conditions, so to speak, seeking out mates. And the question is, how does it happen? What's the so-called brain of the cell, or or eye of the cell. So there's both signal detection and signal processing. And where is that in the cell? And his argument is that this is done by the centrosome, which is composed of two centrioles, and that brings the question of microtubules into the fore. You see here a dividing cell with the centrosome roughly in the middle in green, next to the nucleus in red, and the nuclear wall in yellow. And so he kind of talks in terms of anthropomorphic concepts, discussing, the membrane being kind of like the skin of the human being, the outer layer of our tissue. Then the actin filaments in the cortex, cell cortex represents sort of muscles of the cell performing or contributing to the movement of the cell motility, generating force, and then microtubules performing many functions, but it would be kind of like nerves of the cells, and this we'll explore later on today and next week. We'll take a close look at microtubules, and of course the nucleus has the DNA, the genetic material, the memory of the cell. Now the centrosome in the middle is the most intriguing, the most poorly understood organelle in the cell. It's highly symmetric, and according to Albrecht Bühler, it actually has the capability of detecting electromagnetic radiation with great precision regarding the directions, both in plane and out of plane. And that is what he explored in his work over something like 3 decades or so. There's a lot of material in this website showing actually cells in a somewhat, I would say, intelligent manner, moving towards the source of light. He explored different wavelengths of light, and there was a bead. Light was generated by lasers scattered of the bead, and was supposed to mimic the presence of another cell. So we'll get to it in a second, but this was quite conclusively documented, and there is the sensitivity of this movement toward the source of light as a function of wavelength, and the peak is somewhere in the near infrared, not quite visible, but there is also movement of these cells to visible in the visible range of reflected light from the bead. He isolated all possible chemical effects, and so there's no chemistry, no hormones or biomolecule signaling. It's entirely electromagnetic. So this brings the question of whether it's possibly a quantum effect, and also 2 aspects of it, which is if cells, let's say, forget the bead, but it's supposed to be a replacement for the cell to make it a controlled experiment where you change the wavelength. If this were 2 cells, one is sending signals, the other is receiving. Number 1 question in this is, what part of the cell is sending signals, electromagnetic waves, and the second is what is receiving. And his conclusion is that the easiest part of that two-part question is the reception. He gives excellent arguments in favor of centrioles being the receptors of electromagnetic radiation due to their intricate architecture and the Venetian blind type of arrangement of triplets of microtubules that allow, and they're movable, they allow a very precise determination of the location of the source of light. The question of the origin of emission, if in the experiments, of course, you use lasers, but where does light come from in the living cell? And there are several possibilities, one of which is a byproduct of mitochondrial activity. Mitochondria involve proton pumps and passage of protonic species through the cytochrome has been argued to lead to the emission of a photon. So a living cell would emit photons. A cell which is dead would not, because, well, or a cell that doesn't use mitochondria, but that's a separate issue. So that's kind of interesting and still an open question, not quite resolved. Although these papers that Albert Buehler published, he published in very reputable journals, and so there's a body of literature indicating that cells do indeed exhibit somewhat deliberate phototoxic behavior, and there may be quantum effects in terms of emission. That emission would actually I wrote a paper on showing with my close collaborator in the past, John Dixon, how the activity of mitochondria would lead to this quantum emission of photons. So that's kind of an interesting link to today's topic, which is actually known quantum biology effects today. Before actually, we'll come back to microtubules. I just wanted to show you microtubules in dividing cells. So they perform many functions. The centriole has 9 triplets of microtubules in these arrangements, and the central doublet structure. The whole thing is like a cartwheel, and then so that's a very special organelle. Now microtubules perform other functions, and we'll come back to microtubules later today when we discuss potential information processing inside the cell, information transfer, very intriguing phenomena, not completely understood or explored. I'm actually in the middle of a project which is funded by the Templeton Foundation to elaborate on this experimentally. We have experiments going on at Princeton with Professor Greg Skulls in chemistry, performing tryptophan fluorescence measurements, and I'll come to it in a few minutes. And then the second set of experiments involves delayed luminescence of microtubules exposed to light. So there are some very fascinating functions. I do not, even with 4 lectures, I don't have time to explain all of it, but I will touch base and give you some key points, highlights of this work. So photosynthesis, and by the way, before I talk about this, I want to mention in this connection the importance of it. I think in the beginning I said that quantum biology only so far can only be linked rather strictly to several experiments. So one might criticize it as really spotty, but these experiments and these effects, which are known to be quantum now, are super important because, for example, photosynthesis is at the basis of energy transfer, transduction in the food chain from the solar energy all the way through plants and animals to us. But another connection with physics is that photosynthesis is very much like the photoelectric effect, which, remember, in the first lecture I mentioned was one of the first examples of quantum physics effects, and Einstein got Nobel Prize for explaining it. So here we have light ejecting electrons from a surface, and these electrons are forming a current when subjected to electric fields. And so likewise, in photosynthetic systems, you have light being absorbed, the energy of light or photons is being absorbed and utilized to energize electrons and propel the excitations to collection centers, where this is now transformed into biochemical reactions. So the second experiment, the second, not experiment, but the second quantum biology area that I'll discuss later today as well is quantum metabolism. So this will involve mitochondria, and I will talk about things that are very similar to what I mentioned in the context of solids. If you remember specific heats of solids, the Debye theory of solids versus Einstein theory of solids. And it's also similar to the black body radiation explanation by Planck, the very first experiment. So we have a very close parallel between, physics 120 years ago and biology now in the 21st century, where these two groups of experiments seem to be indicating something very profound happening. Now, the Frolic coherence, which I talked about last time, is very similar also in terms of laser action, which is another macroscopic quantum effect in physics. And then on Tuesday, next week, I will talk about sort of quantum consciousness or possibly quantum anesthesia and so on. So that's kind of similar to a transition from a superconducting state of metals or conductors to a normal state. In other words, the generation or destruction of quantum condensate, which anesthetics might be accused of doing. So there are some interesting conceptual parallels. All right, so now with this in mind, let's take a look at photosynthesis. This is a cartoon explaining how the energy of light coming from the sun is utilized by the chloroplast in the leaves of plants and other structures, as you will see. And it's an extremely efficient process, unlike chemical reactions, which are not very efficient, especially if there is a chain of them, the yield goes down dramatically. Here, photosynthesis kind of reaches almost perfect efficiency. And in 2007, Fleming and collaborators published a paper in Nature showing that electronic energy flow in chlorophyll of plant leaves, cells of plant leaves, is a quantum search problem and can be solved by quantum mechanical equations. There are other types of organisms which use light and harvest the energy of light, and that leads to quantum coherence. And this is single-celled algae in Australia, which can survive even at very low levels of light, and can switch off or on this phenomenon during the photosynthesis. Another one is a bacterium from New Zealand. It's called Chlorobium tepidum or C. tepidum. And these bacteria, the algae and the chlorophyll, have one thing in common. They have these light harvesting centers, LHCs, which are shown here at the bottom, with a very symmetrical chemical arrangement or structure. This is a bit more involved representation, or still a cartoon, but a very detailed representation of the so-called FMO complex and C-tabidem. And in red, you have the pathway of the light rays through the bacterial cell and into the light harvesting center. And in the blue, you have the pathways of the electrons, which are excited and provided for with high energy electrons provided for biochemical reactions. So photosynthesis utilizes the energy of light in connection with specially designed light harvesting complexes, very specifically, as you will see in a few slides from now, and then energy is transferred to photosynthetic reaction centers, where chemical energy is stored and transformed into ATP. So electrons are used to turn carbon dioxide into organic compounds. So electrons, energize electrons with the energy of photons from sunlight plus water plus CO2 produce glucose, which produces ATP. And so the efficiency of these processes is incredibly high, close to 99%. And the important part here in terms of quantum processes is the spacing of these chromophore moieties or centers, which is on a very small distance scale, 15 angstroms, which we know is kind of a limiting factor in quantum tunneling between 10 and 20 angstroms. Beyond that, this wouldn't be very efficient. And this is a representation of the complex of 7 molecules, these multi-ring molecules that can absorb light energy and generate electrons in excited states. Actually, these electrons are not only excited, but they are in a collective state. They are trapped in the system of 7 organic multi-ring molecules, and the wave function is shared between them. And this is a relatively long-lived state with hundreds of picoseconds of half-life. And simulations were done on this and demonstrated how the efficiency of this process involves the perfect orientation and the distances between these molecules. Direct evidence for quantum aspect of it came from these experiments in 2007 and later, where 2 lasers were used to excite this system, and the researchers reported evidence of quantum beating, which would be a superposition of 2 waves with close wavelengths. It's a bit like sometimes when the radio is not quite in tune with the radio station, you have interference and the sound goes up and down. So that's the beating effect. And quantum beating is like it, except that it refers to the electronic wave functions, which are superposed with a very small difference in the wavelength. So these 2 lasers were slightly out of tune with each other, and the researchers were able to observe these quantum beating effects. Okay, so that involves entanglement of wave functions of several rather complex organic molecules, and the question is that the only such effect? and we believe it's not. But before I showed you where else we're looking for such effects, I just want to mention that this kind of process is analogous to a quantum search algorithm in quantum computing, where you look for the lowest energy state among many possible alternatives, and the classical search algorithm would be kind of sequential, going from one state to the next and exploring which one has the lowest energy. In quantum search algorithm is more or less simultaneous. The wave function spreads out and finds the energy minimum, which is incredibly fast, and that's why quantum computing is so powerful. So in this case, these are the papers by Mohseni and others which showed that what's actually happening in chlorophyll and such systems containing FMO complexes, the electronic transitions correspond to a quantum search algorithm. And that opens a kind of general question: do quantum search algorithms play a role in other processes? One of the kind of intriguing suggestions is that nobody really knows why DNA uses an alphabet with only 4 letters, namely the nucleic acids, RNA the same, and whereas proteins use alphabets of the size 20. And of course, the two are connected by the genetic code, which we still don't understand where it comes from, and if there's any logic driven by energy considerations, perhaps, or some other fundamental principles. We simply know it exists, and we deciphered it. It's a bit like a Rosetta Stone, but there is no underlying logic coming from physics or even chemistry. But there has been an observation that quantum search algorithms, like the Grover algorithm, are most efficient when carried out for the following databases. So a single search is most efficient on the database of the size 4, and when you have 3 queries in the search, then the database of size 20 is most efficient for the search, and that kind of accidentally or not, is it correlation without causation or something deeper? But it corresponds to the number of the size of the alphabet in DNA and the size of the alphabet in proteins, respectively. So now back to the question of extensions of the ideas for these kinds of collective quantum states beyond FMO systems and light harvesting centers. Well, there is a compelling similarity between these 7 centers in chlorophyll and 8 centers in tubulin, which makes microtubules. We talked about this, and it was actually suggested several years ago by Hameroff and Penrose that these centers, in this case tryptophans, shown here at the bottom as pathways along microtubule surface, may involve some sort of quantum processing, information processing, and again, that could be a link to what I said in the beginning of this lecture, namely Albert Buehler's intelligent cell and the microtubules not being just mechanical structure forming parts of the cell, but also maybe information processing. And at that, if they could be quantum information processing, then that would give a lot of pause for thinking about what the cell is thinking, is actually processing and reacting to stimuli and so on. So this is the network, as you can see, somewhat similar to the FMO network. Here we have 8 tryptophans with the given orientations and distances. The distances between them are in a similar range. Some of them a bit longer distances. Some are on the order of between 1 and 2 angstroms, definitely close enough for quantum energy transfer, maybe resonant energy transfer. So these chromophoric tryptophan amino acids are suitable for something that is similar to photosynthesis, and that's another picture of this network forming pathways along microtubules. And if these pathways are conducive to quantum energy transfer, then we have a very interesting structure. And each of these tryptophans possesses a dipole moment of about 5.5 dBi, and the neighboring ones interact via dipole-dipole interactions. These dipoles, these are transition dipoles. They can be excited by light. Electrons get excited to a higher energy state, actually UV light, I should say. So it's in the ultraviolet range, high energy, and they re-emit, but they can also transfer this energy to their neighbors. And this has been calculated by my former student Travis Craddock, who's presented and published this work in terms of a computational prediction by writing a proper model with a Hamiltonian, quantum mechanical Hamiltonian for these tryptophans, and finding first of all, finding the eigenvalues, the energy states for the entire system, and then finding how long such an excitation could exist before it dissipates its energy. And the findings were compatible with those for photosynthetic systems, namely on the order of hundreds of picoseconds. So that seems like not too long, but for the purpose of energy transfer and very rapid processing, that should be sufficient. These are the results of this work. The diagrams on the right show you the dissipation of energy, or the decay of the wave function as a function of time. Femtosecond is 10 to the minus 15th, so 2000 femtoseconds would be 2 picoseconds. Yeah, these excitations can in some of these tryptophans can stay quite a bit longer. So this is the general question, whether microtubules, whose architecture is very symmetrical, they are composed of 13 protofilaments, each of which could be many micrometers long. So there are thousands of tubulin dimers forming them, and you can see here a cross section of microtubule, which is a ring, and the red squiggles on these rings are positions of tryptophans. As I said, there's 8 of them per dimer. So for a microtubule, you could have tens of thousands of them, and perhaps this could lead to collective excitations if they are excited by a sufficient amount of energy from photons. So that's another representation of this. And a year ago, I've done some additional simulations on it with a student from France, Léa Guessa, which led to some comparisons with experiments that I'm going to talk to you about now. Some of them are ongoing, some of them have been done already, and that leads to the question of tryptophan excitation. So tryptophan is a very interesting amino acid because it fluoresces. It can be excited and then re-emits this fluorescence. There are two other amino acids which can be fluorescently excited, and this is tyrosine and phenylalanine, as shown here on the right. But tryptophan has the highest efficiency of this process and the highest intensity. So, these are the only three actually which could be excited in a range of wavelengths close to the visible ultraviolet, in fact, but the peak overlapping a little bit with visible. The peak is at about 280. Peak of absorption, peak of emission is about 350 for tryptophan. So that kind of gives us a focal point for studying excitations with ultraviolet range of electromagnetic radiation that could be interacting quantum mechanically with these particular amino acids, and not only tubulin but other proteins as well. But tubulin has a lot of these tryptophans, and tubulin is involved in the formation of microtubules that would be very suitable for special functions. Before I show you some more of the results, I want to mention chemical similarities between tryptophan, which is shown here on the left, and some other important chemical molecules. What I want to draw attention to is the indole ring, which is this. And the indole molecule is shown at the upper right corner. It's a 6-member ring with a 5-member ring and an HN group. So tryptophan has this indole structure in it. Melatonin has it. Psilocybin has it. If you don't know what psilocybin is, it's an extract. It's a molecule, active ingredient in so-called magic mushrooms. And then DMT has it. But also serotonin, which is a neurotransmitter, has it as well. And also native auxin, which is an analog of serotonin present in plants. So all of these molecules may be involved in some kind of interesting interactions at the quantum level, especially if you have a microtubule with lots of tryptophans and you add to it, let's say, serotonin as well. Now, these are the ongoing experiments on the tryptophan fluorescence at Princeton University with Professor Scholes and Arad Kalra, my former PhD student. These are time-correlated single photon counting experiments, and the measurement is focusing on these 3 half lifetimes, time constants T1, tau 1, tau 2, and tau 3, and they characterize the emission spectra from excited tryptophans and tubulin and microtubules. We are in the process of gathering data. I'm not in a position to disclose it yet, but the results are going to be published soon. So that's basically a definitive attempt to determine if microtubules are quantum biology structures such as the FMO systems and chlorophylls I mentioned earlier. Now, another set of experiments that is ongoing within this project is the so-called delayed luminescence phenomenon, which is known in biology. There are some algae, I believe, and some other perhaps insects which luminesce over long periods of time. But here that is my collaborator, Dr. Dogari, and his staff at the University of Central Florida in Orlando are exciting microtubules, tubulin, in the range of 650 nanometer, which is visible with low intensity light, and observing the emission in this specially prepared chamber where light is scattered throughout the boundary and absorbed by the tubulin or microtubules in solution in the inside the chamber. So these are preliminary results which show very long-term re-emission effects lasting many seconds, even minutes, and more so for microtubules than tubulin. And the question is, what is the mechanism of it? And if that's a quantum mechanism, that would be quite an amazing time course of such a effect of storing light energy and then re-emitting it over many seconds to minutes. And here are some arrangements. Different samples were taken, tubulin, microtubules, and microtubules with tubulin, and different results were obtained depending on the structure examined. So again, with this Leah Gassab, who was my intern last year from France, we've set up a Hamiltonian very similar to the one that Travis Craddock and myself put together for the tryptophan interactions. In this case, we've added to it also the dipole moment of the entire tubulin, as well as local electrostatic distributions, which can affect the energies, and that resulted in a fairly good correspondence to the experimental observation in terms of the energy of light that was causing delayed luminescence on absorption by microtubules. We got very similar for energy levels as they were found in the experiments in Florida. Now the question may be asked, okay, it's all very good because you are doing experiments in a controlled laboratory setup and providing laser light as the source of photons. What about the cell? Can you extend these conclusions to living cells? And of course, that's a very valid question. We don't know. What we do know is that there are some sources of even ultraviolet radiation coming from within the cell, and these are documented as I mentioned. Mitochondria are sources of biophotons. These, by the way, are called biophotons, very low intensity ones. And then another one is the recombination of reactive oxygen species, which are also byproducts of metabolism. This is not the question of if they exist, but the question is more of, is there enough intensity of these photons to generate non-trivial quantum transitions in tubulin and microtubules? And we don't know that yet. There may be measurements of biophoton intensity, which indicate very low amounts of photon, something like single photons per cell per minute, maybe on this order. So very, very few. But these are the photons which are emitted by the cell. It's conceivable that some of the photons are emitted inside and reabsorbed and never leave this the membrane, cell membrane. They're trapped inside. So so the question then is, if that's the case, what can this do for biology? Well, these tryptophan dipole effects can be linked to interactions with tubulin C-termini, attachment with MAPs, and also ionic currents flowing around microtubules. So that will be more mundane transformations of light energy into conformational changes of proteins, and maybe performing also some sort of computation, quantum computation. We are in early stages of understanding what this could be doing, but definitely might be very powerful from the quantum computing point of view. So in the rest of today's lecture, I'm going to switch gears a little bit and move to something called quantum metabolism. We'll come back to microtubules on Tuesday, so don't worry. We'll see them again in a different context, in the context of neuroscience. Here, quantum metabolism is a theory that was proposed by Lloyd Demetrius something like 15 or more years ago, and it actually is a very close parallel to the Debye theory of specific heat of solids. And what Lloyd Demetrius observed is that there are some similarities, of course, in terms of metabolic activity in vastly different organisms. Energy can be transduced and must be in different types of cells by different structures. Chloroplasts, we looked at chloroplasts in plants. In animals, mitochondria perform very efficient energy transduction work, basically transforming glucose into ATP through a long chain of chemical reactions, and coupled to these reactions in the mitochondrial membranes are physical transformations, electron transfer processes. So these are the different structures, all very regular. In bacterial membranes, energy transduction networks occur. In chloroplasts, we've seen it. Now in mitochondria have two membranes, inner and outer, and in these inner membranes there are protein complexes which participate in electron transfer. So electron transfer is certainly a quantum mechanical process. It's driven by proton diffusion through the membrane, and these electrons actually tunnel over distances compatible with quantum tunneling. So that is a process which is known to be quantum in nature. Whether this leads to something larger than just an incidental observation, that's something that Lloyd Demetrius investigated and concluded that it is indeed a very fundamental aspect. And let me turn the clock back a little bit and look at a historical timeline for metabolism, looking at metabolism from the point of view of perhaps even evolution, but looking at different species. This goes back to some famous names in physics and mathematics and chemistry, Laplace and Lavoisier in 1780, observed that respiration is a form of combustion, which is really what oxidative phosphorylation is. It's a combustive process producing heat, and then fast forward to 1904, Rubner started measuring body size and measuring metabolic rates of domesticated animals, and correlated metabolic rates with body size, so weight basically, and interestingly observed that this is not linear. In other words, it doesn't scale proportionately to the size, but with some exponent. We'll come to it in a minute. And also size scales with lifespan. And then Kleiber in 1940 performed a systematic study between basal metabolic rates and body size across virtually all living creatures, and established these allometric relations. So it's called allometric scaling laws, where W is the body size and Y is the parameter. Several parameters could be used, basal metabolic rate or field metabolic rate or maximal metabolic rate. These are different technical ways of measuring metabolic rate. But in all cases, there are some very interesting scaling relations which are listed at the bottom of this slide, namely for unicellular organisms, the exponent beta, which relates metabolic rate to weight, is 3/4, not 1. And that 3/4 exponent is actually pervasive, although in plants there is a slightly larger variation between 2/3 and 1. For animals, this exponent varies a little bit between 2/3 and 3/4, again with preponderance of 3/4. And the exponent 2/3 would basically lead you to conclude that it has something to do with the surface area, right? Because if you look at how mass scales with linear dimension, then of course it's through volume. Volume is linear dimension to the power 3 cubic. And if you look at the surface scales with the second power of the linear dimension. Let's say for the volume of the spheres, you have 4 pi r squared, r to the power 2, right? So the conclusion would be that it has to do something with the surface area of the body. Well, if we were to perform respiration through the surface of our skin, of course, that would be explainable this way. And there were some interesting theories proposed in about the same time as Lloyd Demetrius did by others, but they looked at basically transport aspects, the transport of oxygen, typically for respiration. That involved fractal dimensions. I don't want to go in this direction because it really has nothing to do with quantum biology. On the other hand, Lloyd Demetrius explained it using quantum principles, exactly mimicking the approach that Debye took to the specific heat of solids. And let me just, this is a close-up of this inner membrane in mitochondria and the electrons, which are basically hopping through the 4 complexes in the membrane, 1, 2, Roman 1, 2, 3, 4, and they are these are triggered by proton movement across the membrane. But that's a bit more. It's only part of a very complex sequence of biochemical reactions, starting with glycolysis, moving on to the citric acid cycle, and eventually oxidative phosphorylation, which takes place in mitochondria. So this last step is the quantum, involves a quantum process. The other 2 are strictly chemical, and they take place in the cytoplasm. Very interestingly, in some cases, the oxidative phosphorylation completion of the reactions does not happen. And importantly, it doesn't happen when we exert ourselves physically and require quick energy production. For example, sprinters, you know, when you run very fast or do some physical activity that requires quick movement, and then of course after running you feel lactate acid in your lactic acid or lactate in your muscles, in your legs and arms, which is a result of this glycolytic switch, as it's called. So it's the process of energy production is faster, but it's not complete and it's less efficient. But if you need it quickly, then you do it. The other, unfortunately, less useful way of switching to glycolysis is in the case of cancer. Cancer cells also undergo this transition from oxidative phosphorylation to glycolysis, which has been known in cancer cell biology as the Warburg effect. Otto von Warburg, or Warburg in German, Nobel Prize winning biochemist, proposed it in 1926, and as one of the major hallmarks of cancer, and it's still true. So Lloyd Demetrius actually analyzed it, and he said that actually glycolysis is not a quantum process. It's a classical process, and it's inefficient, desynchronized, whereas oxidative phosphorylation requires synchronization across the metabolic enzymes in the cell, and perhaps even beyond. We now know that tissues also synchronize metabolism. So he called it quantum metabolism and introduced the concept of metabolic oscillators. Oscillators have cycles or frequency, if you will. So now you see how I'm going with this to quantum physics, because of Planck quantization, which was the very first step in the building of quantum physics. Frequency in the case of physics was electromagnetic radiation. Frequency was proportional to energy, and here electromagnetic energy. And here in biology, in the case of metabolism, Lloyd Demetrius proposed that energy, which is metabolic energy production, or the rate of energy production, in fact, that's the metabolic rate, is proportional to the frequency of these synchronized metabolic oscillators and enzymes. So it's a very direct comparison that links together a lot of earlier successful and some less successful theories, one of which is the chemiosmotic coupling theory for electron transport, proposed by Mitchell in 1970. He got a Nobel Prize for this chemiosmotic coupling. And then I mentioned to you Froehlich, which is basically about coherent dipolar oscillations in membranes. So that brings us to the enzymatic oscillations in mitochondrial membranes. And then finally, the mathematical step linking these two is energy quantization and linking a la Planck, but in the context of collections of oscillators, Debye's proposal to couple the oscillators and look at waves, so propagating waves of oscillations. So that's how Lloyd Demetrius put it together. That's the model involving Mitchell's chemiosmotic theory and Debye's quantum theory of solids. And surprisingly, all of it kind of works nicely and reflects, and I'll show you just the results, reflects on this fundamental empirical observation summarized by Kleiber in 1940, that the metabolic rate P scales with the body size through the exponent beta, somewhere between 1/2 and 1, if you cover all the species from bacteria to elephants. Alpha is the coefficient which depends on the taxa. The exponent beta really depends on the arrangement of these enzymatic oscillators, and it ranges from 1/2 to 1, with the most common being 3/4. So keep in mind these three numbers: 1/2, 3/4, and 1. Actually, 2/3 also. So 1/2, 2/3, 3/4, and 1. And that's basically the major tenet of quantum metabolism. Enzymes are embedded in the energy transducing organelles. They undergo electrochemical oscillations, and they are coupled across the cell and beyond. And quantization principle introduces the energy linked to the frequency. And that's the paper that actually introduced that concept by Demetrius in 2003, Quantum Statistics and Allometric Scaling Laws, Scaling of Organisms. So you even have quantum in the title. And he made analogies, formal analogies between physics and biology. So let's say thermodynamic parameters, such as in physics, temperature is controlled, and then you measure heat capacity like the Dipole, and you can also introduce entropy, and because heat is related to entropy production. In the case of biology, temperature does not change much. I mean, most of the organisms are constant temperature organisms, including ourselves. And so temperature is replaced by cycle time. And the cycle time basically is inverse to the frequency, how fast these oscillations occur. Heat capacity is replaced by the metabolic rate, and thermodynamic entropy by entropy production rate. Okay, so that's the next hypothesis, that the metabolic rate is proportional to the cycle time to some exponent beta, which in this case I think was empirical adjustments. And I will very quickly just show you, without really asking you to follow the equations too closely, but the audience on YouTube might be physics trained or mathematical trained. They could take a look at these equations, and this basically is the way Debye calculated the average energy for a collection of oscillating entities with quantization, and then you calculate the total metabolic energy stored in the network by summing it up, or integrating. If you have enough of these, you can take the frequency into the continuum limit, and there is a maximum frequency, just like in the Debye theory of solids, and obtain two extreme cases to the quantum limit with the scaling of the average energy with the cycle time to the power d+1. D is the dimensionality of space. Importantly, we'll come to it in the next slide. And then the classical limit, where there is a linear proportionality between the average energy and the cycle time. So now, this is at the bottom, you actually have the results for the metabolic rate and body size. So in the classical case, you have proportionality, which almost never happens. It's the limit of this exponent beta being 1. In some cases it is. So maybe I'll just mention one more interesting tidbit about this. So this is the so-called classical limit of metabolism, where oscillators are decoupled. And that would be actually physiologically relevant when you study cancer, because cancer cells are not synchronized. They desynchronize from themselves and desynchronize from the tissue of origin. And therefore, that would be one very interesting application of this theory. That's in the extreme classical case. Let's say cancer cells would be much more energy dependent, or hungry, or because they don't scale with the fractional exponent. In other words, the larger the tumor size, the greater the demand on the energy supply to the tumor. Whereas for the rest of the body, we are much more efficient due to synchronization. And this is the quantum limit, where you have the scaling of the metabolic rate with the mass or the weight to the power d over d plus 1. So d is dimensionality. And okay, now we are in this quantum limit. So what does it mean? That if d equals 1, if it was a, let's say, a snake, a quasi-linear structure, or some worm, then you would have d equals 1 over 1 plus 1, so 1/2. And the scaling of the power, not the metabolic rate, power is energy per time, so that's metabolic rate, is proportional to the square root of the weight of that snake or worm or some linear creature. Now, not many of these creatures could be found, but let's say if you go to two-dimensional, let's say organism plants, then so flat surfaces, then the scaling goes with d equals 2. So d over d plus 1 would be 2 divided by 2 plus 1, which is 2/3. That's the second exponent I wanted you to remember, and indeed you get it here. So for quasi two-dimensional systems, biological systems, metabolic rate would scale with the 2/3 exponent of their weight. And finally, when d equals 3, so we would consider ourselves three-dimensional biological systems and most of the animals and so on, then you have d equals 3, and 3 divided by 3 plus 1 is 3/4. This is the dominant exponent in all the empirical findings. And what does it tell us? Well, it tells us that our metabolic demand does not increase linearly, it's sublinear. That is, if you increase the size of the organism by a factor of let's say 16. 16 is 2 to the power 4. If you enlarge something 16 times, let's say, you compare a child to an adult with this kind of ratio of mass, then the metabolic rate or food consumption would increase only 8 times, not 16 times. So you see, you have a great advantage as you grow a large organism, because they become more synchronized and more efficient. There's division of labor, and the cells become quite efficient working together. And that's why I told you that in the case of cancer, that's not the case. That's not the situation. There, the metabolic demand increases with the size of the tumor. So I'm almost done, and I'm kind of, this slide is very busy. I don't want to belabor the point, but it gives you all the numbers that kind of I didn't mention before, because I wanted to convey the concepts. But here are the numbers that are interesting in this context. You know, we produce, so energy in the human body is produced largely in the form of ATP molecules. This is the currency I mentioned, and I think in the first lecture, it's quantized. So we use, you know, integer numbers of ATP molecules, not half. So that's the energy quantization per se. But we produce roughly the amount of ATP equal to body weight every day. That doesn't mean that we create them from nothing, but they are created from ADP in a cyclical process. But the turnover of the amount of ATP is staggering, and that is equal to 10 to the 21st ATP molecules per second for the average human. That's an astounding number. And then we have about 3.7 actually times 10 to the 13 cells in the human body. So, and each cell has about 1,000 to 2,000 mitochondria, which produce ATP. So there are approximately 3.5 times 10 to the 16th mitochondria in the human body. And then when you do the division of this, you get about approximately 30,000 ATP production events per mitochondrion per second. That's also quite an incredible turnover, right? So 30,000 ATP molecules being produced in an average healthy mitochondrion in every second. So then the question of efficiency comes into the picture. So ATP, we need glucose and other, but glucose is, let's say, the easiest to discuss. One glucose molecule gives rise to 38 molecules of ATP through mitochondria, oxidative phosphorylation. On the other hand, in glycolysis, only 4 ATP molecules are produced, so it's almost 10 times less efficient. And that explains, so although it has nothing to do with quantum biology, but I think it has to do with the incredible burden cancer puts on the human body. Because when you now think about two effects put together, the first one, that cancer cells do not cooperate, so the size of the tumor is proportional to the power demand that it has, or the metabolic demand. And the second thing is that this demand is exacerbated by the fact that the cancer cells produce energy in an inefficient way, 10 times less efficient if it's completely glycolytic. Having said that, they are not entirely glycolytic, but even if you switch to glycolysis for 50% of your energy demand, then you suddenly see the problem. The problem can be such that the ravenous consumption of glucose by cancer cells can overwhelm the human body, the rest of the body, even if the tumor is, let's say, 1 or 2 kilograms in weight, and that leads to well-known and tragic physiological effect of cachexia, muscle wasting, and the late-stage cancer patients lose a tremendous amount of weight. And actually, this is one of the also early signals that something is not, if it's an involuntary weight loss, that quite often has to do with this process. So that's the classical metabolism according to Demetrius. Quantum metabolism is efficient, is scales sublinearly, and uses oxidative phosphorylation. So I'm not going to go too much with the rest of the slide, but I will just point out two things. Typically, the oxidative phosphorylation reaction is approximately 1,000 cycles per second for each complex. So it's about 1 millisecond time duration, or that's the characteristic time scale. And then Yeah, there is something intriguing here because you can go back to the Max Planck quantization principle, which we started this whole series with. And I told you that the Planck constant is a fundamental constant in nature, is equal to 6.6 10 to the minus 34 joules per second. Joule seconds, not per second. However, if you do the same calculation for the human metabolism, you end up with a constant which is like the Planck constant. I call it the biological Planck constant, and it's actually about 11 orders of magnitude larger than the physical Planck constant, which is intriguing because it kind of corresponds to the number of atoms per mitochondria cell of influence. If you have 1,000 mitochondria, you figure out how many atoms are in this cell and how many atoms per mitochondria in the cell. That's roughly the range. So in other words, it kind of makes sense to talk about a biological Planck constant, but there is something maybe not as fundamental because it depends on, as always in biology, which cell, and so on. But nonetheless, it is energy is proportional to frequency. So yes, and this is a teaser for Tuesday. I finished today's lecture, but I would like you to come back on Tuesday, and we will talk about these brain questions in the context of quantum biology and things like memory storage, computational power, information processing. And actually the first question will be probably last: what is consciousness, and can consciousness be understood with respect to quantum physics, quantum biology, maybe something else. So thank you for your attention, and if you have questions, please go ahead. I'll stop sharing.
Okay,
If I can find. Yes.
there we go. All right, so let's try this immersive thing again. All right, there we are. So any questions? Comments? Tasmin raised hand. Yes, Tasmin.
Hello. Okay, so a lot of the second half of the lecture was a little too much for me, so I'll have to watch it again. But in the beginning part, when we were talking about the mitochondria, so is that implying that mitochondria have the potential to be luminescent, like bioluminescent? And is that also applicable to bioluminescence in the animals that we know of, like fireflies and like jellyfish?
Yes. Well, I'm not the expert in bioluminescence per se, but I think from what I know and looking at mitochondrial action, the emission of photons or light from mitochondria is like a byproduct of its activity because of the charge transfer. When you create electromagnetic waves, and photons or light are examples of electromagnetic waves, you just need two opposite charges, namely plus and minus, which is a dipole, to oscillate. When two opposite charges oscillate, you emit electromagnetic waves. Now, the frequency of these waves, of course, depends on the rate of oscillation of these dipoles. So to go back to your question, that's how I see. This is a difficult question because the intensity that we observe these biophotons at is so low that you have to have ultra sensitive equipment, and there's environment you have to shield and so on. So I'm not quite sure if anybody has actually measured directly if mitochondria emit light, and if so, at what rate. But I would expect so. And maybe next time I'll bring up a slide which shows you the biophoton emission from different living systems. I have it. I didn't prepare this time, but I can show you next time what intensities are typical for light emitting living. Well, we all emit some of it, but very low. I was actually visited a biophotonics lab in Prague in Czech Academy of Sciences, and I saw that they had a device with very sensitive photodetector, and you put your hand in this specially designed glove, and they measure the amount of light that you emit from your hand, from the surface of your hand, which they did for me. So, but these are small amounts, and you have to have very good equipment to see it.
Thank you.
You're welcome.
Yeah, you know, this question of whether humans really have an aura, you know, something surrounding us. If you're carrying a cell phone, of course, you do have a sort of periodic aura where your phone is you know, connecting to the tower and that sort of thing.
Yeah.
Yeah.
Well, we definitely emit infrared, like every object at a high finite temperature. We emit infrared, but this is not visible, and that's why, you know, in combat, night combat, you know, you have these infrared goggles that you can see.
Yeah. You
can see the enemy soldiers or whoever you're fighting from far away because of the emission of infrared radiation. So that's electromagnetic waves, but in a wavelength which is longer than visible.
Yeah. You know, in the recordings from this course, one of the striking things is in the early days, the cell phone sounds were quite a prominent artifact in the audio track. And we just don't find that anymore. Yeah.
Really?
Yeah.
Well, like cell phones not ringing, just being there. I mean,
Yeah, yeah. So
interfering with the sound.
Yeah, you would be it somehow got incorporated in the audio system from the room, and you could hear these extra sounds from the cell phones. That was a feature of this course, let's say from 2011 to 2015, something like that, with the recordings that we were making, which were otherwise very, very high quality. So it had all sorts of rules about cell phones and, you know, putting your, I think it's still in the rules for the course, but you have to put your cell phone in airplane mode and stuff like that. But it doesn't really seem to be operative today. So it's one way in which the world has improved, I guess.
Yeah. So anyway, I apologize if this was too technical, but I think this is one of the necessities that sometimes you have to dig deep into the you know, the technicalities to at least for some to be able to follow with some of the mathematical and physical basis for it. It's not a simple math that you need here, but nonetheless, I hope it makes sense. And as you said, it's worth listening to it. There's also literature or references you can read.
What I've always felt is when the students know that they're not required to know the formulas for the exam, then it becomes part of the art. You know, formulas are sort of the part of the art of science, right?
Yes, and I don't feel we should require this because it's an interdisciplinary course.
Sure.
But I think that the main thing at the end, at least, that I wanted to emphasize is this aspect of energy efficiency related to synchronization between cells. And it's a topic that I believe will be very important in the future in terms of diagnosing diseases early, and maybe even related to aging, where the efficiencies decline, and you could measure these things quite accurately and predict the onset of diabetes. And I think at the end of this series, I'll mention a few things about disease, which is related to this transition from quantum to classical.
Yeah. So, there's something interesting about the next lectures after yours. Shauna Pandya is sort of a superstar. She's a very inspiring person to be in the same room with. But I need to figure out if she's going to be presenting by Zoom or in the actual room. And that might have a big effect on how many students we end up with in the room.
Is she from the U of A?
Yeah, she is. She's in the front lines of fighting COVID. She works in local emergency rooms.
Wow.
She's been to nearest space. She's been weightless. She's sort of done everything, And
oh my God, I feel like I don't belong in this club.
No. You're both cutting edge, but in different ways.
Thank you.
And
You're very kind.
she's a little bit younger than you are. So anyway, that's what's coming next. Yeah.
Great.
Okay. Well, thank you very much for this, and we'll see you on Tuesday.
Thank you so much. See you next week.
Right.
Bye-bye.
Bye-bye.